Okay, so welcome to this next video in which we are discussing the opioid receptors. Okay, so we're currently discussing the pathway downstream of a ligand binding and activating an opioid receptor. And we've discussed that all four of the opioid receptors are coupled to GI, heterotrimeric G proteins, which means that the heterotrimeric G protein has an alpha subunit that's either G-alpha I1, G-alpha I2, or G-alpha I3. Now we've seen the effect of the alpha I subunit, that it will inhibit adenylyl cyclase 5 and 6 and stop the activation of adenylyl cyclase 1, 3, and 8 via calcium, thereby resulting in a lower cyclic AMP level within the cytoplasm of the cell. But we're now looking at the effects of the beta-gamma subunit. We've seen that beta-gamma subunits can bind to these G protein activated potassium channels, and when that happens, what will happen is the G protein gated potassium channel will open and allow a net movement of potassium ions out, okay, which will cause hyperpolarization of the electrical potential difference across the cell membrane. What we now want to discuss is the effect that the beta-gamma subunit also has on voltage-gated calcium channels. Okay, so we're going to start off with a discussion of the structure of voltage-gated calcium channels, because they have quite a complicated structure. There are many different subunits that all join together to make a voltage-gated calcium channel. Okay, so I will first start by drawing a sort of three-dimensional picture of what a voltage-gated calcium channel looks like, and then we'll look at the in-detailed structure of the different subunits. Okay, now voltage-gated calcium channels are often abbreviated to VGCCs for short, for voltage-gated calcium channel. Right, so let's show one of these voltage-gated calcium channels in the cell membrane. So let's let this represent the two layers of the cell membrane, okay, and then what we will have is the main portion of the voltage-gated calcium channel here, okay, and this is the portion which actually allows calcium ions to move through it, and therefore really is the actual pore-forming portion of the channel. Okay, so basically, it is called the alpha-1 subunit, and it is actually all one protein, and we'll have a look at the membrane-spanning topology of this protein in a moment. Okay, now, that's not all that you generally have to a voltage-gated calcium channel, however. You have what are known as auxiliary or accessory subunits. Okay, so for instance, you have an alpha-2 delta subunit, like so, which sort of binds onto the alpha-1 subunit. And this consists of two separate proteins which each come from a common precursor protein. Okay, so what will happen is there'll be one gene that codes for both of these subunits, and this portion here is the alpha-2 portion, and this portion that straddles the membrane is the delta portion. Okay, there's one gene that codes for both of these portions. It will be transcribed into a piece of mRNA, and then that mRNA will be translated to produce one massive great protein, and then that massive protein will be chopped into two to make the alpha-2 and the delta subunit, and then the alpha-2 and the delta subunit are stuck back together in a different way to the way that they were originally joined by being in one massive great protein. And this makes what's known as the alpha-2 delta subunit here. Okay, right. That's one of the auxiliary subunits. You also have sitting over here next to the alpha-1 subunit a gamma subunit. Okay, and also on the cytoplasmic side, bound to the alpha-1 subunit, you have a beta subunit. Okay, so let's start off then with looking at the actual membrane-spanning topology of each of these proteins. And I want to stress once again that this is supposed to be the pore here through which the calcium ions flow. And in fact, if you take away all the accessory subunits, the alpha-1 subunit alone will form a voltage-gated calcium channel. So these accessory subunits are truly accessory. They just change the properties of the alpha-1 subunit slightly. Okay, so let's now discuss the membrane-spanning topology of the alpha-1 subunit. Now this is a big protein, okay? It's generally around 2,000 amino acids long. Okay, so let's have a look at its membrane-spanning topology. So basically, you'll start off with the amino terminus over here, okay? Then it will span the membrane once, span the membrane a second time, third time, a fourth time, a fifth time, then it will try to span the membrane, fail, and that will be called a p-loop. Okay, and then you'll have the sixth membrane spanning alpha helix like this. Okay, then you repeat this same structure 3 more times. Okay, so again, here's another repeat of it. 5 membrane spanning alpha helices, a p-loop, and then the 6th membrane spanning alpha helice. Then a loop, then another repeat of it. Okay, 5 membrane spanning alpha helices, a p-loop, and a 6th membrane spanning alpha helix. Then at the final repeat of this, then we have 5 membrane spanning alpha helices, a p-loop, and then a 6th membrane spanning alpha helix, and then we have the carboxylic acid tail down here. Okay, now these 4 repeats that you have, they are called domain 1. So this first one is called domain 1, the second one is called domain 2, this third one is called domain 3 here, and this final one over here is called domain 4. Okay, so you have domain 1, domain 2, domain 3, and domain 4. Now if you look at any one of the domains, i.e. let's take domain 1, then there are names for each of these membrane spanning alpha helices and the p-loop. Okay, so basically the first membrane spanning alpha helix is called S1. The second membrane spanning alpha helix will be called S2. Okay, so let me try and give an arrow to this. And then it goes on. The third one will be S3, the fourth one will be S4, and the fifth one will be S5. This loop here between what's S5 and then what's S6 here. Okay, S6 is this final one. This is called the p-loop, okay? And this nomenclature works for all of them. So what you would say is, I'm talking about, well, let's say if I wanted to talk about this membrane spanning alpha helix here, I would say domain 3, S3, okay? So that would tell me which domain it's in, and then when I know which domain it's in, I would then know which membrane spanning alpha helix of that domain I was talking about, okay? So the naming S1 to S6 and then the p-loop goes for all 4 domains. Okay, so basically these 4 domains wrap around one another. Well, they wrap around to form a pore, okay? So it will be like so. Basically, you can imagine it being split into these 4 domains, although it is one massive protein, this massive thing here. Okay, so let's imagine splitting it up into these domains. So this would be domain 1 here. This portion at the back will be domain 2. This portion over here will be domain 3. And this portion at the front here will be domain 4. Okay, so you have these 4 domains sort of together forming this pore, basically, and that's the alpha-1 subunit. Okay, so basically there are multiple genes for alpha-1 subunits of voltage-gated calcium channels, okay, and this determines the main electrical properties of these voltage-gated calcium channels. Okay, specifically there are actually 10 genes for alpha-1 subunits of voltage-gated calcium channels, okay, that produce 10 different, slightly different alpha-1 subunits that do have different conductance properties and activation properties. Overall, we can sum it up as electrophysiological properties, okay? And again, when I say 10 genes, I mean if you take 1 copy of each of the homologous chromosomes. We're not counting each gene twice, basically. If you took account of the fact that you have 2 copies of each chromosome, then you'd have 20 of these genes, 1 on each of the homologous chromosomes. Okay, right. So 10 genes coding for 10 different subunits. Okay, so what are these? Well, basically, we group them into families, and there are 3 families that we group them into. And we group them into the CAV1 family, the CAV2 family, and the CAV3 family. Okay, for voltage-gated calcium channel 1 family, voltage-gated calcium channel 2 family, voltage-gated calcium channel 3 family. Okay, now what are the members of the CAV1 family? Well, in the CAV1 family, you have the gene CAV1.1, you have the gene CAV1.2, you have the gene CAV1.3, and you have the gene CAV1.4. And all of these genes code for then a protein that will have the membrane spanning topology like so, and will fold up into an alpha-1 subunit that forms a pore down the middle, and those will then be called the CAV1.1 subunit, the CAV1.2 alpha subunit, the CAV1.3 alpha-1 subunit, and the CAV1.4 alpha subunit. Okay, and basically if you make a voltage-gated calcium channel with one of these subunits from the CAV1 family, then your voltage-gated calcium channel is called an L-type voltage-gated calcium channel, an L-type VGCC. Okay, so that is what is meant when people say, I'm talking about an L-type voltage-gated calcium channel. They mean that the alpha-1 subunit is within this CAV1 family, and it's one of these 4 genes here. Okay, let's turn our attention now to the CAV2 family. So the CAV2 family contains CAV2.1, CAV2.2, and CAV2.3. Okay, now basically, if you make a voltage-gated calcium channel with a CAV2.1 alpha-1 subunit, then that voltage-gated calcium channel is called a PQ-type voltage-gated calcium channel. Okay, so if someone says they're dealing with a PQ-type voltage-gated calcium channel, that means that the alpha-1 subunit is a CAV2.1 gene. Okay, if you use the CAV2.2 subunit as your alpha-1 subunit, then that will give you an N-type voltage-gated calcium channel. Okay, finally, if you use CAV2.3, that will give you what's known as an R-type voltage-gated calcium channel. So basically, what type of voltage-gated calcium channel you have, the naming depends on which alpha-1 subunit you have. Now, if it's any of these ones in the first family, it's just called an L-type, but all of these members of the second family have separate names, basically. Now, in the third family, there are then again 3 genes, okay? CAV3.1, CAV3.2, CAV3.3. Which produce the proteins CAV3.1, CAV3.2, and CAV3.3. Now, if you make a voltage-gated calcium channel that has as its alpha-1 subunit any of the 3 genes within this third family, then that voltage-gated calcium channel is called a T-type voltage-gated calcium channel, so a T-type VGCC. Okay, what we will do is we'll continue our discussion now with the gamma, the beta, and the alpha-2 delta subunits, and then we'll come back to, once we've finally finished talking about voltage-gated calcium channels, we'll come back to the effects of the beta-gamma subunit on these voltage-gated calcium channels. So, let's now turn our attention firstly to the beta subunit, okay? So the beta subunit has a structure with 4 membrane-spanning four alpha helices, but they're not membrane-spanning alpha helices. And basically, the beta subunit is going to bind to this intracellular loop between the first domain and the second domain here. Okay, so I'll show the beta subunit here. So here are these 4 alpha helices, like so. Okay, so here's the amino terminus of this beta subunit, and here's the carboxylic acid terminus that just fits in there. Okay, so basically these 4 portions here, which I'm now highlighting in turquoise, these are alpha helices. You have 4 alpha helices in this beta structure, okay, and they all sort of fold up like this, and then this beta subunit then binds to this loop between domain 1 and domain 2. Okay, right. So, how many different genes do you have for beta subunits? Because this gives you another level of complexity. So, okay, beta subunits, you have 4 different beta subunits. You have beta 1 gene, the beta 2 gene, beta 3 gene, and then also beta 4 gene. Okay, now you have 4 genes, but you do actually have more than that beta subunits, and it's the same reason as we had 16 genes for alpha subunits of heterotrimeric G proteins, and then 21 subunits. So some of these will have multiple splice variants. For instance, beta 2, for instance, has the 2 splice variants beta 2A and beta 2B. So if you see letters after the subscripted number, that's just, whoops, this should be beta 2B. That just denotes which splice variant you're talking about. Okay, so there are multiple different beta subunits that can then bind to your alpha 1 subunit. So this is increasing the diversity of voltage-gated calcium channels that you can then produce. Okay. Right, so we've talked now about the alpha 1 subunit and the beta subunit, and I should just mention the beta subunit can alter the properties of the alpha 1 subunit. It can alter the conductance, it can alter what sort of voltages it will activate at, things like that. Okay, let's next turn our attention to the alpha 2 delta subunit. Okay, so basically, if we get another piece of paper for this, okay, right. The alpha 2 portion is completely extracellular, whereas the delta portion has this membrane spanning portion. Okay, so let me show this. So basically, if we have the plasma membrane like so, you'll have the delta portion, which will have a single membrane spanning alpha helix, like so. Okay, so this is the delta subunit of the alpha 2 delta subunit. And then you'll have the alpha 2 portion, which will be here. Okay, so let's just draw the alpha 2 portion, like so. Okay, so it has a few alpha helices, like so. Okay, right. So, basically, hopefully what you can see is that the alpha 2 portion is a portion which is completely extracellular, whilst the delta portion is this membrane spanning portion. Okay, so this should be the C terminal of the alpha 2 portion. So this is alpha 2 here. I'll highlight them in separate colours. So we'll have delta in turquoise here, and then we'll have alpha 2 in pink here. And basically, they are held together by disulfide bonds that are going to form between the alpha 2 polypeptide and the delta polypeptide. Okay, so they're held together by disulfide bonds. Okay, now this subunit will also then bind on to the alpha 1 subunit. Okay, and again, they modulate the function of the alpha 1 subunit. Specifically, the alpha 2 delta subunit is thought to be very, very important in controlling the location of the voltage-gated calcium channel, i.e. directing it to certain portions of the cell membrane, basically. So, for instance, in neurons, the alpha 2 delta subunit is thought to be important in potentially locating the voltage-gated calcium channels nearby docked synaptic vesicles, basically, so that they can open and release calcium onto the machinery that docks the synaptic vesicle, and that can then trigger the exocytosis of that synaptic vesicle. Okay, right. So there are 3 different alpha 2 delta subunits, okay, encoded by 3 different genes. So remember, both of them are made on the same precursor protein and therefore by the same gene. So there's 3 genes for alpha 2 delta subunits, and these are alpha 2 delta 1, alpha 2 delta 2, and alpha 2 delta 3. So there's quite a bit of variation there. So there's another place where you can increase the diversity of voltage-gated calcium channels that you can potentially produce. Okay, and finally we'll finish off by looking at the gamma subunit. So what's the structure of that gamma subunit? Okay, so remember, let me just get the picture back. The gamma subunit sits alongside the alpha 1 subunit. And basically the gamma subunit has 4 membrane-spanning alpha helices. So it has its amino and its carboxylic acid termini on the cytoplasmic side of the membrane, like so. And then it has these 4 membrane-spanning alpha helices here. So this is the gamma subunit. Okay, now not much is known about the gamma subunits which are available for voltage-gated calcium channels. One of them that is widely studied is the stargazing subunit, okay, which was found in a type of mouse that had a mutation in this protein, okay, and this subunit does associate with the alpha-1 subunits of voltage-gated calcium channels. Okay, so this is an example of a gamma subunit, but of all of the accessory subunits, the gamma subunit is the least well understood. Okay, right, so we'll conclude that video there, and in the next video what we'll do is look at the effect of the beta-gamma subunit on these voltage-gated calcium channels.